This is a lecture on bacterial transduction for April 15th, 2020. And the main goals of this lecture are just to define what transduction is, and then compare the function and the mechanisms of generalized and specialized transduction. And so, as we talked about in the previous two lectures, bacteria need to use several different mechanisms to generate genetic variation because they can't do so through sexual reproduction. And so bacteria have used mutations as well as horizontal gene transfer to increase their genetic variation or genetic diversity. And we have talked a bit about conjugation and transformation, which are two types of horizontal gene transfer. And today we're going to focus on transduction, which is the third. And what transduction is simply the transfer of genetic material between two bacterial cells by a virus. And so we sort of use this virus as a transfer mechanism to take a gene from a donor bacterium and then transfer it into a recipient. And transduction typically results from an error that happens during the virus's life cycle. And transduction is common in nature. It's particularly common in aquatic or marine environments, where the amount of viruses available to transfer genes between two bacteria is quite high. And so just to review the lytic and lysogenic life cycles of viruses, bacteriophages, or viruses that infect bacteria, can sort of have two options in terms of life cycles and infecting their bacterial host cells. And so bacteriophage that enter into the lytic cycle will insert their DNA into a bacterial cell, and then directly following the insertion of that DNA will replicate the DNA as well as make all of the proteins necessary to construct new virus particles or virions, which can then be released from the cell and cause it to lyse or die. And then those bacteriophages can go on and infect other host cells. And that's the lytic cycle, right? So it's an immediate response that results in lysis and release of virus particles. However, there are some bacteriophage that can enter into a different life cycle, which you can see here on the right, which is known as the lysogenic cycle. And in the lysogenic cycle, a bacteriophage will still inject its DNA into the host cell, but rather than multiplying that DNA to create new viruses, it will integrate its viral DNA into the bacterial chromosome, which you can see here in blue. And this piece of viral DNA inside the bacterial chromosome is known as a prophage. And the prophage DNA can last in a bacterial chromosome and be replicated and passed on to any daughter progeny cells. And in this way, viruses can sort of lay dormant within a bacterial host cell for many years until an event induces them or triggers them to cut out that prophage and then enter into the lytic cycle, where that viral DNA can be replicated, packaged into new virions, and then released from the cells. And so this process of transduction, or this transfer of bacterial DNA via viruses, has two different mechanisms of transduction. There's a generalized mechanism and a specialized. And so in terms of generalized transduction, it occurs only during the lytic cycle. So when a virus is immediately infecting, replicating, and then lysing cells. In addition, generalized transduction can transfer any part of that bacterial genome to a new recipient cell. And that's because the main hallmark of generalized transduction is that the bacterial DNA is mistakenly packaged into viruses during the assembly phase to create what's known as a transducing phage that can bring this DNA to a new cell. And so when a bacteriophage infects this bacterial cell, as you can see on the top, the viral DNA is entered into the cell, and the host DNA, that bacterial chromosome in blue, is degraded into smaller pieces. And then as these pieces of DNA are multiplying and new viruses are being assembled here, There is an occasion where some pieces of bacterial DNA in blue get packaged mistakenly into a new virus particle. And then this guy who has bacterial DNA inside of him is known as a transducing phage. And rather than infect new cells, what this transducing phage can do is bind to a new host cell, and insert this bacterial gene into the host, where it can recombine with the host's chromosome through the process of transduction. And so as you can see here, we have a bacteriophage attaching to a host cell and then inserting its viral DNA. That viral DNA can replicate, and the bacterial chromosome is degraded, and then new virus particles are assembled, some of which contain viral DNA in pink, and this one guy here, which contains a bacterial piece of DNA in dark blue. This is your transducing phage, which can now effectively travel and bind to a new recipient cell, insert its DNA. This bacterial DNA can be recombined into the host cell's chromosome. So in addition to this generalized transduction mechanism, there's also a specialized transduction that doesn't occur during the lytic cycle, but occurs during the lysogenic cycle of a virus. And in specialized transduction, only specific parts of the bacterial genome are transferred. And as a result of improper excision or the improper cutting out of the prophage during induction, or the transition from lysogenic cycle to lytic cycle. And so when a prophage integrates into the bacterial chromosome in blue, it does so at very specific sites in the bacterial chromosome. And because this viral DNA integrates at specific sites, when it's cut out, even mistakenly, we already know the sequences of DNA, and there's only very specific ones that would come along as a result of an improper excision. And so in the case of specialized transduction, you have a temperate phage, which has integrated its viral DNA into the chromosome, and then after induction starts to proceed, rather than just the prophage itself in red being cut out, some bacterial genes come along for the ride as well. And in addition, some viral DNA is left behind in that host cell chromosome. And after this excision of this sort of hybrid bacterial viral DNA piece, replication takes place, and many, many copies of those small viral bacterial pieces of DNA are made. And they are then incorporated into the assembling virions or phage particles. And as you notice here, each single one of these phage particles contains some viral DNA in red and some bacterial DNA, which makes every single one of the phages generated from this mechanism a transducing phage, or a phage that's capable of transferring bacterial DNA to a new cell. And so once these virions are released, they can bind to new cells and insert their viral/bacterial DNA, which can be integrated into the genomes of those recipient cells. And what's important to remember about this is that because not all of the viral genes from the prophage make it into the new virions, these new virions are not really considered effective. They lose their virulence, they lose their ability to quote-unquote infect new cells, and the recipient cells cannot generate new virus particles, right? And so transduction is not particularly helpful to viruses because recipient cells that are infected by this bacteriophage can't help propagate that virus any further because they don't have all the genes necessary. But transduction is helpful to bacteria because it does allow transfer of genes from one bacterium to another, which is not something that would be possible through sexual reproduction. And so it's a way for this bacteria to increase its genetic diversity. And so here during specialized transduction, you can see this bacteriophage attaching to the cell, inserting its DNA into the cell, or it can be integrated into the bacterial chromosome to form a prophage. And then when induction happens, or it transitions from lysogenic to lytic cycle, this piece of DNA is removed. It should be all purple, meaning all viral, but it does, as you can see here, contain some bacterial DNA. And it's then replicated, packaged and assembled into new virus particles, which are then released from the cell. And every cell, every virus, as you can see here, has a little bit of bacterial DNA in it, meaning that every virion is a transducing phage particle. And then that transducing phage can bind to a new recipient cell, insert this viral bacterial mixed piece of DNA, and then that can be integrated into the genome of this host cell.